Hello, everyone. So I'm Chiari. I'm a computer scientist who works on artificial intelligence, and I lead the AI for Health Institute here at WashU. So my mission is to apply AI to all kinds of medical and healthcare problems. And I'm fortunate to have the opportunity to work with Dave and the team to co-lead this project. And in particular, talk about our recent progress on Project 4, which is to phenotype Chiari malformation type 1 with AI. You have heard several times today that Chiari is a heterogeneous disease, and there's growing recognition that it's likely to be a constellation of clinical phenotypes rather than a singular disease entity. Hence, a key goal of this project is to identify distinct and clinically meaningful phenotypes based on a combination of data-driven methods such as AI, as well as clinical insights. Of course, we are fortunate to be able to exploit the Pathways registry, by which, at the time of analysis, comprises over 1,000 patients from 42 medical centers. And you can see this in this initial analysis that's led by Shan Gupta, who is a resident at neurosurgery, and my doctoral student, Ziqi Xu. So this is typical multidisciplinary research combining AI with clinical expertise. So in this analysis, we're looking at these clinical data, radiographic data, with the goal of identifying different phenotypes, which in data science lingo would be means clusters. If you look at this multidimensional data space, you're trying to look for what subset of the data points are clustered together. So then hopefully they represent distinct clinical phenotypes of this disease. So the modeling approach, first we split the 42 centers data into the internal and external cohorts, 21 centers each. This is to test reproducibility, which is very important in AI analysis that I'll get to later. And then, I want to emphasize one thing before I forget. So if you look at the combination of all these different modalities of data, we basically end up with over 500 variables. This just highlights how challenging it is to phenotype Chiari malformation, because human is not very good at looking at 500 different variables and figure out, this is subtype 1 versus subtype 2, which is too high a dimension space to look into for humans. But this is something that AI is particularly good at. AI is really good at looking at a lot of data in many, many dimensions of data and then identify patterns, in this particular case, clusters. In particular, methodology-wise, we first do a feature selection. This is first using statistical and AI methods to identify 28 variables that are particularly important and effective in separating the data into different clusters. Then we also want to make sure we capture the clinical expertise that's acquired over years and in the literature. So we also, through clinical surveys of the Pathways consortium, identify 5 additional variables that's included in the clustering analysis. Then we apply unsupervised K-mode clustering. If you know anything about clustering, such as K-means clustering, K-mode is basically K-means clustering applied to discrete, so binary data variables. So the end results, I'm happy to report, we were able to identify 3 distinct clusters, with this AI-driven method. I'll walk you through this figure to your right. So I'll first just look at the one on the right, which is the so-called radar plots. You see the variable names around the circle. These are different variables that were selected using the feature selection methods. Then you'll see 3 different color shapes within the circle. These represent the 3 clusters identified using AI. So where, of course, the points on boundary of these shapes represent the mean value of that group of patients of that particular variable. So intuitively, you look at these shapes that are very, very different, right? So these represent very different characteristics, right, in this multidimensional data space. And if you look at the figure to the left, right, that's basically the same data, same results as plotted differently where the variables are lined up vertically. Again, you see the three curves representing three clusters that are very distinctive from each other. Now, also, what is very important is we redid this clustering, right? So that in the external cohort, recall we split the medical centers right into two groups. Right, on top is one group of 21 medical centers. On the bottom is the other 21 medical centers. We repeated the clustering on both the internal and external cohorts. You realize right, the clustering results are very similar to each other. This is truly exciting and reassuring in a sense. It seems like we have identified right, clinical phenotypes that are generalizable right, across patient populations. This is clearly still the initial findings, but certainly look very promising, right, both from a data standpoint and to a clinical standpoint. Right, so we published this result in neurosurgery right, earlier this year. And from a clinical standpoint, right, so these are the specific three different clinical phenotypes identified through clustering. Right, the first is basically the classic CM1 plus series patients whose primary complaint is about headaches. The second cluster, the green one, are patients with multiple neonatal medical issues, but few classical symptoms. Right, whether they will develop those symptoms remain under study. And finally, the blue cluster, cluster 3, are Chiari patients with syringe-related spinal effects. Right, this is particularly important because early diagnosis and intervention can help prevent major surgeries. So to summarize, what we have found to date was certainly typical radiologic definition is inadequate right, for heterogeneous disease like Chiari malformation, and these identified clusters potentially assist a more precise disease management that are tailored to different phenotypes. Thank you.